Okay. Good afternoon. My name is Ying Zhang. I'm a fourth-year graduate student at Stanford Burnham. It is my honor today to present you our research, which was published last year about a three-dimensional structural view of the central network of Thermotoga maritima. For hundreds of years, people have been searching for ways to living cell. The search started from the development of first microscope and was taken further since he discovered the connections between phenotype and genotype. Since the 1970s, as development of new sequencing techniques, especially the last decades, many hyperpotent developed. Now we are equipped from which we can start to build a complete picture cell. Molecular level study of a cell, there are many different areas. Among them are structural biology and system. Structural biology usually focuses on the three-dimensional structure of micromolecules in the cell. Those 3D models to look for the functionality of and how they are regulated by other molecules in the living. Whereas systems biology aims at building the between different molecules and using mathematical modeling the connections between different molecules. These two fields are very important, helping the cellular mechanisms, but the connections between are vague. Usually given a structure, we do not know what's in the cellular networks, whereas giving a not know their three dimensions and how they perform the entire function of a living cell. Our research, we propose an integrated view of systems and structural biology. Our idea is that structural biology would provide additional information about the protein functionality and how they are regulated by small molecules, where system biology will provide a bench where we could organize all different types of structures so that we could start to look in a larger scale on how the structures are distributed in the network and how they function together to sustain the life. The model organism we selected is maritima. It is a hyperthermophilic bacteria isolated from the thermal vent in the ocean. This organism likes to live in very high temperatures. The optimal growth is on 80 degrees centigrade. It is one of the fully sequenced bacteria, contain very small number of genes, only around 1,800. So it's a manageable system for us to work with. It's also very branched in the bacterial lineage, so it gave us some insight into the evolution of bacterial species. The most important thing is that it has the potential to produce hydrogen from organic waste. It's really very important because today we're in lack of energy. The three-dimensional reconstruction of Thermotoga maritima contains 3 steps. The first is data collection and mathematical modeling, and second is network, and the third is structural modeling and mapping. On the first step, we collect all types of data from various resources, including computational predictions and also literature search. And also, our other information comes from genome context and functional context analysis, and we combine all this information with the physiological data published today in the literature. Based on those informations, we build gene-protein reaction associations, which basically connect each gene with their encoded proteins in turn is connected to one or a couple of reactions that happen in the cell, which is catalyzed by this enzyme. And then, based on the GP associations, we built a mathematical model, which is this matrix where it connects every reaction with the participating compounds. From this matrix, we have to make simulations on the functionality of the cell. After the first step, we will have a draft network of the metabolic system. And so in the second step, we use a bunch of different simulations to try to look at the network and see whether they are accurate enough and represent the state of art of our understanding on this bacteria. The first thing we looked at is precursor generation, which are precursors. We make sure that the precursors can be generated so that certain important compound exists in our model organism. And the second part, we looked at biomass generation to make sure that the live and can produce enough biomass to sustain growth. And we also look at the gap regions in a mathematical model, and if they have gaps, we go back to the first step, either to look at the literature or using other more advanced computational method to try to fill the gaps with new additional reactions or additional gene annotations. And another thing we looked at is the ATP generation and energy production so that the cell can produce enough energy to sustain itself. The last thing is physiological data comparison, where we make simulations based on our mathematical model and then compare the published physiological data and see whether they correlate, at least qualitatively. After the second step, we got a model that is complete enough and collect all the information that's available today about the bacteria. And the metabolic network is defined as complete because they could sustain life as what was observed in experiments. And then we start to build structural models and map them into the network to build a three-dimensional view. We build structural models based on three different approaches. The first is experimental methods. Thanks for the efforts in protein structural initiatives and especially Joint Center of Structural Genomics. This Thermotoga maritima genome is now covered with more than 300 different protein structures. So we have a large amount of data structures we could work with and try to map them into the metabolic network. For those that haven't been experimentally determined, we run them through a high-throughput homology modeling process where we take each of the sequences and try to search for a best template and build a structural model based on the template, and then using scoring and other modeling evaluation approaches to choose a model that could best represent the three-dimensional feature of a protein. For those that failed this high-throughput approach, we try to go back and using manual approaches or some other approaches to build supervised models so that it's as accurate as we can make. This pie chart here only shows you what are the proportions of each of the methods that help us cover the entire metabolic system. Here, the slides just show you some statistics on the scope of our reconstruction. Basically, we covered around more than a third of the entire Thermotoga maritima genome, and each of them is determined with X-ray crystallography or high-throughput structural modeling. Only 22 models we are built based on topological, their topological features because they are transmembrane protein and we couldn't map them, find a good template for them. I emphasize that our current reconstruction only represents our state-of-art understanding of the bacteria. It's not a final version, but it's the best that we can do in the current situation based on our understanding of this bacteria and all the experimental and computational methods available. And this reconstruction is an iterative process. As we can gain more knowledge from additional experiments or additional computational methods, we might extend this work to make them more accurate and complete. And so now we have a three-dimensional model of the metabolic network. What can we do with it? The first thing we did is to look at gene essentialities. But since we have a mathematical model, we could do as much experiments as we want. So we used this model to make gene deletion simulations. We tried 1,000 different rounds of gene deletions, which successively delete individual genes until the cell cannot sustain life anymore. So based on those deletion experiments, we define each of the genes in Thermotoga maritima metabolic network into three groups. They are core essential genes, which are genes that always have to exist to sustain the production of biomass. And the second group is synthetic lethal genes, which are proteins that sometimes are needed, but not in all the simulations. And the third group is non-essential genes. They're genes that always are not needed using a given nutrient condition. So basically what we suggest is that the core essential represents the branches in the network that has to exist during the nutrient take-up and utilization process, whereas synthetic lethal genes represent the branches in the metabolic network, so that when in a random process, if this branch is selected, it is needed, so it becomes essential. But if it's not selected, it becomes non-essential. So it has this binary feature, sometimes essential, but not all the times. And the non-essential genes are simply not needed for taking up this nutrient we're giving. Based on this classification, we can look at their three-dimensional topological features of the proteins that compose each of the three categories. We see that core essential genes is composed of larger number of protein families, but each family have less representatives. So here, the family we're looking at is the protein fold families defined by scope. And the non-essential genes contains smaller number of protein families, but each family contains larger number of representatives. To make it clear, we can see it from the cumulative curve of the families. Here we can see that the largest family in the core essential, which is blue, is much smaller, maybe half the size as the non-essential families. What does this tell us? I think this means that the core essential genes contain some three-dimensional fold species, so they have to be present in the core of a metabolic network, whereas the non-essential proteins are produced by duplication of the core essential, but specification to bind other compound. Therefore, the core essential is a core where they have a number of fold species that shall always exist in the metabolic network, where non-essential is the extensive side, which help the organism to take new nutrients and utilize new resources. The second thing we looked at is functionality of proteins versus their position on the metabolic network. To do this, we grouped each reaction pairs into 3 groups. First is similar reactions. They're reactions that carry out similar chemical mechanism. And the second groups are connected reactions, simply adjacent in the metabolic pathways. And the third groups are unrelated reactions. They are neither similar nor connected. And based on those, we compared their chances, the chances of the protein that catalyze either similar, connected, or unrelated reactions of them to share a fold or not. What are the chances? We discovered that enzymes catalyzing similar reactions having 10 times more chance to share a fold when compared with enzymes that catalyze connected reactions. This means that enzymes were duplicated to recruit recruited to new pathways. And this supports the batchwork model proposed by ISCB, which basically say that the enzymes, when they are duplicated, the chemical mechanisms are conserved, but they develop new binding site to utilize new compounds. Surprisingly, the connected reactions still have 10 times more chance to share a fold than the unrelated reactions, which means that local duplication will play some role in the evolution of metabolic networks. To summarize, the integrations of systems and structural biology is very useful in studying cellular networks. I hope I convinced you so by presenting you the analysis we've done so far on Thermotoga maritima. It helped us to understand the function of proteins and how they're distributed in the network. This so far is very useful, but keep in mind, in the beginning of my talk, I was saying that we want to build a complete picture of the entire cell. But now, we're still far from that goal. If we look at the entire Thermotoga maritima genome, about a third of them encodes metabolic proteins and genes. And another third encodes processes like information storage or like signaling processes and so on. And the rest is largely uncharacterized or unknown. So how can we start to extend our current version of metabolic reconstruction and make them more complete at first, and then incorporate other processes like regulations and information signaling processes? If we take a closer look at the metabolic part of the Thermotoga maritima genome, we covered around 80% of the entire metabolic part, but the energy production process and carbohydrate metabolism processes are less known in today. However, if we look at the bright side, thanks to the structural genomics, we now have over 50% of the proteins encoded in Thermotoga maritima genome already having a very high quality structural models. So in the next step, how to utilize those models will be essential for annotation of the entire genome and try to incorporate them in the reconstruction. Therefore, for the future directions, we would like to combine the structural approach with sequence information for protein function predictions. How would we do that? Since we already have a final version of the reconstruction, they have structures and perfect annotation on the functionality of proteins. It provides us a very nice data set from which we could start to derive knowledge And we can use those derived knowledge to try to extend our annotation. So our plan is to work with both protein families and non-homologous proteins. For protein families, we would like to look at proteins in the same family and see what are the features that determine their specificity and functionality, so that we could help generate accurate predictions for the rest of the family. And for non-homologous proteins, we observed that in the metabolic pathways, there are adjacent proteins which belongs to different families, but they bind the same chemical compound. So why was that? What makes them? What features are common between them so that they could bind the same thing? So we would like to making those type of analysis so that we could get a workable tool for predicting other proteins. So with this, I want to end this advertisement of my poster. If you want to know more about the additional analysis we're doing on follow-up of our current project, please visit the poster V09. I already presented it, but if you're still interested, please stop by or just be there so I can show you what we are doing currently. Hopefully, I would like to acknowledge people who involved in this work. I want to thank our collaborators in UCSD, in Dr. Bernard Paulsen's group, and also people in our group who are involved in structural analysis and structural modeling, and also the experimental structural biologists who helped solve the structures we use in this project. And especially thank my advisors, Adam Gosick and Andrew Austin. Last but not least, thank the funding resources, travel fellowship, and our research. Thanks all for attention.
Thank you very much, and that's already the first question.
So you showed that enzymes that carry out similar reactions are 10 times more likely to share the same fold than enzymes in neighboring reactions. Mm-hmm. And neighboring reactions are 10 times more likely to share the same fold as enzymes in unrelated reactions. Mm-hmm. So are enzymes in neighboring reactions overrepresented for the similar reactions, and did you correct for that?
So the question was, are enzymes that are adjacent in the pathways share the same fold?
No, if I could imagine that two enzymes that are neighbors in a pathway would also have the same chemistry. So they would fall into both classes of being neighbors and carrying out the same type of reaction. Is that the case?
I got your question. So the question was, does adjacent reaction also belongs to the group of similar reactions? The answer is mostly no, because we did manual creation after the automated classification. We looked at each of the pairs that was classified as both connected and similar. There are only, I remember, 4 or 5 cases like that.
Okay, so there's no need to correct. So that doesn't explain your correlation between folds then?
Right. I think we need to correct that because we have around 340 different reactions, and the pairing is like a lot more than only 4 or 5.
Another question there.
Hi, thanks for an exciting talk. I was wondering, in your data, do you find any evidence to the concept that enzymes from the same metabolic pathway form some sort of or molecular machine that would act especially at the same place doing reactions of the same metabolic pathway?
You mean the multiple complex enzymes?
Right.
You mean in which analysis?
From your data.
Yes. We have cases in our metabolic reconstruction where we need more than one enzyme to carry out the same function. But in the statistical analysis, we remove the cases like that because they're too complicated to, in terms of counting the fold sharing pairs. So we just remove this part of data.
No, I mentioned more enzymes that carry out adjacent reactions than the same.
Adjacent reactions, do they form complexes?
Yeah, whether they do or not.
I think they are, but we don't capture it in the model.
Thank you.
Thanks.
Another question?
Those 44 odd exceptions you found.
The reaction gaps?
The reaction gaps, yes. How many of those can be attributed to possible misannotations within the genome?
Can be attributed to
Misannotation.
misannotations.
Yes.
This, I don't have an estimation, but I do have a list of these 44 reactions. They're generally reactions that we think should exist in the organism based on some literature or some physiological data. There could be errors with the 44. For example, some reactions may not be one reaction in this specific, they might instead composed of a new pathway. And also, sometimes we do not fill the gaps just because we don't have a proper gene that is known to fill the gap. We don't have enough information. As I mentioned, this version of the reconstruction is pretty initial version. So if we use more scrutinized method, maybe we can hopefully fill maybe at least half of them.
Okay, thank you.
Thanks.
Thank you very much.
Thank you.